Hey everyone, and welcome to the Art Biolab channel. Today, we're diving into something truly revolutionary, one of the most incredible scientific tools ever discovered, CRISPR. And get this, it's not just about reading the code of life, it's about having the power to literally rewrite it.
Just think about that for a second. What if we could edit the very source code of life itself? We're not just talking about reading a genetic blueprint. We're talking about going in with surgical precision and rewriting it. That's the mind-blowing promise of the tech we're going to unpack today. All right, so here's our game plan. First, we're going to go back to its origins. And you might be surprised to learn it started as a natural defense system in bacteria. Then we'll see how scientists had that aha moment and turned it into a gene editing tool. After that, we'll get into the nitty-gritty of how we can delete and even add genes. And to top it all off, we'll explore its coolest trick, controlling genes without cutting them at all. Okay, first up, where did this game-changing technology even come from? You might think it was born in some fancy, futuristic lab, right? Nope. It actually comes from an ancient, microscopic war, the constant battle between bacteria and the viruses that hunt them.
Yeah, CRISPR is a bit of a mouthful, but the idea behind it is just brilliant. So imagine a virus attacks a bacterium. The bacterium fights back by literally snipping off a tiny piece of the virus's DNA. And then, this is the clever part, it stores that little snippet in its own DNA. It's basically building a genetic most wanted gallery of past invaders. Think of it like a collection of viral mugshots. So if that same virus ever shows up again, the bacterium knows exactly who it is and can take it out immediately.
This is where things get really interesting. Scientists looked at this bacterial defense system and had a stroke of genius. They thought, wait a minute, if this system can be programmed to find and cut a specific piece of viral DNA, could we maybe reprogram it to find and cut any piece of DNA we want? And the answer, of course, was yes. Okay, so the whole system really boils down to three main components. First, you've got your workhorse, the Cas9 protein. The easiest way to think of it is as a pair of molecular scissors, just ready to cut DNA. Then you have the guide RNA, or gRNA. This is the brains of the operation. It's like the GPS coordinates that we program to tell the scissors exactly where to go. And finally, there's a little safety feature called the PAM sequence. The Cas9 scissors won't make a cut unless they see this little code right next to the target. It's like a permission to cut signal. So putting it all together, how does it actually work? It's really just three simple steps. Step 1, find. The guide RNA zips along the DNA, leading the Cas9 protein until it finds the perfect matching sequence. Step 2, bind. Once it finds the spot and sees that little PAM sequence, Cas9 locks on tight. And step 3, cut. The molecular scissors do their job, making a clean, perfect slice right through the DNA.
Okay, so we've made the cut. Now what? Well, this is where the real magic happens. The cell, you see, freaks out when its DNA is broken and immediately kicks its own natural repair crews into gear. And it's by cleverly hijacking these repair processes that we can do some incredible things, like turning genes off or even adding new ones.
So the cell has two main strategies for fixing broken DNA. Think of the first one as the quick and dirty fix. It's an emergency response. It just grabs the two broken ends and smashes them back together. It's fast, but it's sloppy, and it often makes little mistakes. Then there's the high-precision pathway. This is the careful, meticulous one. It finds a perfect template and uses it to repair the break flawlessly without a single error. So, let's say we want to disable a gene. How do we do it? We actually take advantage of that messy, error-prone repair system. We tell CRISPR to make a cut, and then we just let the cell's clumsy repair crew do its thing. It rushes in, makes a mistake, and that tiny little error is often enough to completely scramble the gene's instructions. It's like a typo that makes the rest of the sentence total gibberish. The cell can't read it anymore, and boom, the gene is knocked out.
Okay, but what if we want to do something more sophisticated, like add a new gene or fix a faulty one? For that, we use the other repair system, the precise one. Here's how it works. Along with the CRISPR scissors, we sneak in a new piece of DNA, a donor template. This template has the exact code we want to insert. So when the cell's high-precision repair crew shows up, it sees the cut, finds our template, and uses it as a blueprint to fix the damage, perfectly stitching in our new genetic code. That's what we call a knock-in.
But wait, there's more. CRISPR isn't just about cutting and pasting. This is where it gets really, really cool. Scientists figured out a way to tweak the system so we can control how much a gene is being used. Think of it like a dimmer switch or a volume knob for your DNA. And the best part is, we can do it without making any permanent cuts at all.
The trick lies in a modified version called dCas9.
which stands for dead Cas9. And it's dead because scientists basically went in and broke its scissors. They tweaked the protein so it can't cut anymore. So now, it still uses the guide RNA to find its target and bind to it, but instead of cutting, it just sits there. It becomes a programmable anchor that can clamp onto any DNA sequence we want.
And why is an anchor useful? Because we can attach other things to it. By sticking different proteins onto this dCas9 anchor, we can create that dimmer switch. For example, if we attach a protein that acts like an accelerator, we can tell dCas9 to go to a gene and turn its activity way up. We call that CRISPR-A for activation. Or we could attach a brake protein to turn a gene's activity way down. That's CRISPR-I for interference. We can fine-tune gene expression without ever touching the underlying code.
And the power of this is just incredible. I mean, check this out. Scientists can take a regular old adult cell, say a skin cell, and use this CRISPR activation system to turn on just a handful of specific genes. And by doing that, they can reprogram the cell, winding back its clock and turning it into a powerful stem cell. A cell that can then become almost any other type of cell in the body. The potential for regenerative medicine here is just staggering.
Now, of course, a breakthrough like this doesn't just happen overnight. It was built on the shoulders of giants through years of patient scientific work. Back in 2005, they figured out what Cas9 even did. A couple of years later, they understood the guide RNA's role. And then in 2011, the final piece of the puzzle clicked into place, showing how it all worked together as this elegant, programmable system. So, in what feels like the blink of an eye, CRISPR went from being this quirky defense mechanism in bacteria to a complete all-purpose genetic toolbox. We can knock genes out, we can knock new ones in, we can dial their volume up and down. It's really true what they say. For the first time in history, we're not just reading the book of life, we're finally learning how to write in it.
And that, of course, leads us to a pretty massive question. The potential is almost limitless, right? We're talking about everything from curing genetic diseases to designing crops that can feed the world. But this kind of power, well, it comes with a huge amount of responsibility. We're standing at the beginning of a whole new chapter in biology. And the question really is, for all of us, what story should we write next?